Welcome to StudentPal. In this Grade 12 Biotechnology lesson, we are beginning Chapter 3, Gene Cloning. Gene cloning is not simply the multiplication of DNA. It is an experimental reasoning process in which a biological question is converted into a carefully designed construct, introduced into a suitable cell, and tested until the identity of the recovered clone is established. By the end of this lesson, you will be able to plan that process from candidate gene identification and nucleic acid isolation to enzymatic construction, DNA delivery, screening, and independent confirmation. Keep your notebook ready, pause whenever you need to study a diagram, and let us build the chapter step by step. Here is everything we are going to learn. We will begin by asking how a candidate gene is identified and what evidence connects it to a biological trait. We will then follow the isolation of usable DNA or RNA and learn how yield, purity, and molecular integrity affect every later reaction. We will examine restriction endonucleases, sticky and blunt ends, DNA ligase, polymerases, reverse transcriptase, phosphatases, and kinases. Next, we will design a recombinant construct, compare directional and non-directional assembly, and examine chemical transformation, electroporation, conjugation, microinjection, liposome-mediated delivery, and particle bombardment. We will distinguish selection, screening, and confirmation, compare southern, northern, and western blotting, master the 3 stages and controls of PCR, and choose between genomic and complementary DNA libraries. Finally, we will interpret diagnostic evidence and organize full-mark CBSE answers. With that roadmap in mind, let us begin with the central question of the chapter. Imagine a plate containing dozens of colonies after a cloning experiment. Every visible colony has survived the selection condition, but that does not mean every colony carries the desired insert. Some may contain an empty vector. Some may contain the insert in the wrong orientation. Others may contain a rearranged or mutated construct. Gene cloning therefore works as an evidence funnel. We begin with many possible cells, enrich those with a selected vector-associated property, screen the survivors for the expected insert, and finally confirm identity, orientation, junctions, and sequence. Throughout this lesson, keep one question in mind: what direct observation supports each claim, and what alternative explanation still remains? That habit separates a plausible cloning result from a scientifically verified clone. A candidate gene is a sequence suspected to influence a trait, pathway, or product. The candidate may emerge from biochemical evidence, inheritance patterns, mapping, sequence similarity, expression data, or comparison of contrasting organisms. But association is not the same as function. If a sequence is abundant in disease-resistant plants, that observation supports a relationship, yet resistance may be caused by another linked gene or by a regulatory change. A strong hypothesis predicts what should happen when the candidate is cloned, expressed, disrupted, or transferred. It also specifies comparison groups and measurable outcomes. In an exam answer, move through 3 levels: evidence that identifies the candidate, a molecular plan to obtain it, and an experiment that tests its function. Gene cloning can produce the material needed for that test, but cloning alone does not prove the gene causes the phenotype. Nucleic acid isolation begins by disrupting cells or tissues while protecting DNA or RNA from degradation. Detergents can disrupt membranes, salts and enzymes can help separate proteins, and purification steps remove lipids, polysaccharides, phenolic compounds, nucleases, and other inhibitors. RNA isolation requires especially careful control of ribonucleases. The final sample must be judged, not merely collected. Concentration indicates yield. Absorbance ratios can suggest contamination, but they are not a complete quality test. Gel electrophoresis can reveal fragmentation, degradation, or unexpected forms. Why does this matter? Contaminants may inhibit restriction digestion, ligation, PCR, or sequencing, while damaged DNA may remove part of the target. Preserve sample identity with clear labeling, include suitable controls, and match the isolation method to the organism, tissue, and downstream purpose. Good cloning begins with a molecule that is both present and usable. Restriction endonucleases recognize particular DNA sequences and cleave phosphodiester bonds in the DNA backbone. Some cuts create single-stranded overhangs called sticky ends, others create blunt ends. Compatible sticky ends can base pair and often support efficient joining, but the enzyme choice must be based on the complete map. A useful enzyme should cut at intended boundaries without destroying the insert, origin, marker, or required expression signal. Two different enzymes can create directional ends, reducing the chance that the insert enters backwards. Reaction conditions also matter. Buffer composition, temperature, DNA methylation, enzyme amount, and excessive incubation can affect specificity. A partial digest leaves uncut molecules, while unwanted activity can create unexpected fragments. The correct exam explanation connects recognition site, type of end, construct design, and expected diagnostic fragments instead of merely naming an enzyme. Cloning requires a coordinated set of enzymes. DNA ligase forms phosphodiester bonds and seals compatible DNA backbones. DNA polymerases synthesize DNA and may be used for amplification, fill-in reactions, or end modification depending on the enzyme. Reverse transcriptase copies an RNA template into complementary DNA, allowing expressed eukaryotic sequences to be cloned without their genomic introns. A phosphatase removes terminal phosphate and can reduce vector self-ligation. A kinase adds phosphate when a ligation strategy requires it. These enzymes are not interchangeable. Their substrate, direction of reaction, and required cofactors determine their role. A useful way to answer an exam question is to organize them by task: obtaining a DNA copy, preparing compatible ends, preventing an unwanted product, joining fragments, and confirming the final molecule. Always state what changes chemically, not only the name of the enzyme. Construct assembly begins with a map of the intended final molecule. Vector and insert are prepared with compatible ends, mixed under suitable conditions, and joined by ligase or another defined assembly strategy. Directional cloning with two different ends can control orientation, but it does not automatically confirm the reading frame or sequence. The insert must avoid disrupting the origin selectable marker and required regulatory elements. If protein expression is intended, promoter direction, translation start, reading frame, stop signal, and any tag junctions must be checked. Controls make the result interpretable. A vector-only ligation estimates background caused by self-closure. A no-ligase control can reveal uncut vector. After transformation, the number and type of colonies are observations, not proof of the desired construct. The design map predicts expected fragments and PCR products. Molecular tests establish which molecule was actually recovered. DNA transfer methods solve different physical and biological barriers. Chemical transformation can make bacterial cells competent to take up DNA, while electroporation uses a controlled electric pulse to create temporary membrane permeability. Conjugation transfers DNA through cell-to-cell contact. In animal systems, liposome-mediated delivery packages nucleic acid in lipid structures that interact with cell membranes, while microinjection places material directly into a cell or nucleus. Particle bombardment accelerates DNA-coated microscopic particles into plant tissue and other difficult targets. Method choice depends on cell type, cargo size, efficiency, cell damage, equipment, stable versus transient outcome, and biosafety. A delivery event is only the beginning. The DNA must persist or integrate as intended, and the cell must remain viable. Compare methods using the same criteria, rather than presenting an unrelated list. Selection, screening, and confirmation. Answer different questions. A selectable marker allows cells with a vector-associated trait to survive under defined conditions, enriching the relevant population. Screening then distinguishes likely recombinants from empty vector or incorrect candidates. Methods may include insertional inactivation, colony PCR, probe hybridization, or diagnostic restriction digestion. Every screen has possible false positives and false negatives. Confirmation uses stronger evidence, commonly sequencing the insert and its junctions, sometimes supported by independent restriction or PCR results. State the direct observation before the conclusion. A colony growing on antibiotic is an observation supporting selection. A PCR band of expected size supports the presence of a region between two primer sites. Neither alone proves the complete sequence. A high-quality cloning workflow therefore moves from broad recovery to targeted screening, and then to independent molecular confirmation. Blotting methods share a general logic. Separate molecules, transfer them to a membrane, and detect a selected target. Southern blotting examines DNA. Restriction fragments are separated, transferred, and detected with a complementary labeled probe, allowing questions about fragment size, copy pattern, or integration to be investigated. Northern blotting examines RNA and can provide evidence about transcript size and abundance under defined conditions. Western blotting examines protein, usually using a primary antibody for target recognition and a label detection system. The three methods are not interchangeable because DNA presence, transcription, and protein production are different claims. Band position, intensity, and specificity must be interpreted with molecular size markers, positive and negative controls, and suitable loading comparisons. A band is a direct observation. Identity and biological meaning are inferences supported by the detection system and controls. The polymerase chain reaction amplifies DNA between two primer-defined sites. During denaturation, heat separates the template strands. During annealing, the temperature is lowered so primers can bind to complementary target sequences. During extension, a thermostable DNA polymerase synthesizes new strands from the primers. Repeating these stages can produce an approximately exponential increase in the intended product, while reagents remain effective. Specificity depends strongly on primer design, annealing conditions, and template quality. The product length is determined by the primer positions, not by the polymerase choosing a gene. PCR can help screen colonies, obtain an insert, or test a diagnostic junction, but a band of expected size may still contain an incorrect sequence. For full credit, draw the three stages in order, label primer direction, and explain how repeated cycles increase the target region. A good PCR experiment is designed with controls before the tubes enter the machine. A no-template control contains the reaction components, but no sample DNA. Amplification here suggests contamination or primer-derived products. A positive control contains a known amplifiable target and shows whether the reagents and cycling conditions can work. An extraction control can reveal problems introduced during sample preparation, while an internal control can help distinguish target absence from reaction inhibition. If the sample gives no band but the positive control works, consider missing target, poor extraction, inhibitors, or primer mismatch. If multiple bands appear, consider non-specific primer binding, unsuitable annealing temperature, or excessive magnesium and cycle number. Gel bands must be compared with a molecular size marker. The strongest conclusion combines correct controls, expected size, and when identity matters, sequence confirmation. A DNA library is a collection of cloned fragments designed to represent a source of genetic information. A genomic library is prepared from genomic DNA and can include exons, introns, regulatory regions, intergenic sequences, and repetitive DNA. A complementary DNA library begins with RNA, commonly messenger RNA, which is copied by reverse transcriptase. It therefore represents genes expressed in the sampled cells, tissue, condition, and time, and usually lacks introns. Choose the library from the question. To study gene structure or regulatory DNA, a genomic library may be required. To obtain an intron-free coding sequence or compare expressed genes, a cDNA library may be more useful. Recovery depends on fragment size, vector capacity, number of independent clones, genome size, transcript abundance, and screening strategy. A library is a population whose usefulness depends on representation, not merely a box containing many clones. Suppose 3 selected colonies must be tested for a 2 kilobase insert. Colony PCR with primers flanking the cloning site can rapidly distinguish an empty vector from a larger candidate product. A second primer pair joining vector and insert can test orientation. Plasmid DNA from promising colonies can then be cut with diagnostic enzymes. The observed fragment pattern is compared with the sizes predicted from the construct map. Finally, sequencing across both junctions and the insert tests nucleotide identity, reading frame, and unwanted mutation. Each method contributes different information. A correct PCR size does not reveal every base. A diagnostic digest may not distinguish 2 sequences with the same map. Sequencing is powerful, but poor quality reads or incomplete coverage still matter. The best conclusion is based on agreement among independent observations and includes a suitable empty vector or known construct comparison. For a 3 or 5 mark CBSE answer, begin with the purpose of gene cloning and identify the candidate DNA vector and host. Present the procedure in biological order. Isolate usable nucleic acid, prepare insert and vector, assemble recombinant DNA, deliver it to cells, select survivors, screen candidates, and confirm the clone. When an enzyme is named, state its molecular job. When a method is compared, use parallel criteria such as target molecule, mechanism, evidence, and limitation. For PCR, write denaturation, annealing, and extension in sequence and include controls. For blotting, match southern with DNA, northern with RNA, and western with protein. For libraries, connect the source material to the information represented. Avoid claiming that selection proves the insert or that one band proves biological function. A label diagram plus an evidence-based conclusion makes the reasoning visible and earns marks. Close your notes and rebuild the chapter as one evidence chain. Begin with a candidate gene supported by a testable hypothesis. Isolate nucleic acid of suitable identity, purity, and integrity. Choose enzymes from the construct map, prepare vector and insert, assemble the recombinant molecule and include controls for background. Select a delivery method that matches the host cell. Use selection to enrich cells, screening to locate likely recombinants, and sequencing or equivalent evidence to confirm the final clone. Use southern, northern, and western blots for DNA, RNA, and protein questions respectively. Use PCR with correctly behaving controls to amplify or diagnose a defined region. Choose a genomic library for broad genome representation and a cDNA library for expressed intron-free sequences. If you can state the observation, inference, control, and limitation at every stage, you understand gene cloning as experimental science rather than a memorized protocol.